Good day everyone. So today we are going to learn the chapter Biotechnology Principle and then Process. So this is a first KIMS. So a KIMS is a knowledge imparting session. It is of approximately 30 minutes duration. So that first 5 minutes will be get an awareness of what is required to understand the concept. In the next 20 minutes we will learn some of the concepts in the chapter. The next 5 minutes you will answer a question from a old competitive exam question papers. So this session will give you the learning required for a board exam as well as the ability to appear for a competitive exam. So the rest of the content in the chapter will be covered in the KIMS that follows. Thus the chapter will be done in nearly 8 to 10 KIMS. Now let us talk about the prerequisite for the first KIMS. So coming to the prerequisite, the first one is a biotechnology. What is in biotechnology? So biotechnology is nothing but we are going to manipulate the microorganism for the production of an enzymes or a product. So we are going to alter and then we are going to manipulate the microorganisms and then we are going to get products like enzymes or antibiotics or any products. So we are going to get useful products for a human use. The next one is a sterilization. What is the sterilization? The process of removing microbes, which is called as a sterilization process. So if a place which is mentioned as a sterile area means that place will be free from microbes. So then only we can able to do the process of any biotechnology or whatever the process. When we did in a sterilized condition, it will be help us to get a proper result. The next one is a plasmid. What is a plasmid? The plasmid is an extra chromosomal small circular double-stranded DNA in a bacterial cell. So which is present, which is a present extra of a chromosomal element in the bacteria. So this is a normal their genetic bacteria, that is a chromosomal bacteria, whereas chromosomal DNA, whereas this plasmid is an extra chromosomal, which is extra of a chromosome, so which has a small circular double stranded DNA structure. Then next one is a plasmid. Sorry, next one is a in vitro. What is that in vitro? In vitro means when doing some process in a laboratory scale means which is going to be called as a in vitro process. Whereas the process which is happen inside the body which is called as a in vivo. If a particular process which should be happen inside the body means that is will be called as an in vivo process. When we try to do that process in a laboratory manner means which is going to call as an in vitro process. One example will be fertilization process. So usually that normally fertilization will be happen inside the body, but when we try to fertilize the egg and then sperm when in a laboratory manner means which is going to be called as an in vitro fertilization. Then the next one is a vector. So vector is nothing but which is used to carry a one gene or one DNA from a one place to another place. One example of a vector is a mosquitoes. So always that mosquitoes when they are going to bite an infected person means they are going to carry the microbes from the infected person, and then when this same same mosquito when they are trying to bite a normal person means these microbes can be get introduced into that normal person, so that normal person also become a affected person. So this mosquitoes going to act as they are going to carry a microbes from a one person to one another person. So likewise the basically the vector means which is going to carry a information from a one organism into a another organism. So coming to the objectives, we are going to study about biotechnology and then genetic engineering process. So this is one typical representation of a plasmid, which is an extra chromosomal element. So when it is an extra chromosome means the chromosome contains DNA. So the plasmid will be made up of DNA, which is a small and then circular. So the biotechnology which deals with the techniques of using organisms, either it is a live or a enzymes. So in a biotechnology process we are going to use either a live organism or an enzyme which is isolated from a organisms. So we are going to use these two and then we are going to produce a products which is useful for humans. Some of the products which will be made through the biotechnology, through the microbes or an enzyme is a curd, another one is a bread, another one is a wine. So these are a biotechnology or a processed products. So in this they are going to be mainly done by microbes. So they are going to, the microbes going to play a major role. So that is why it is a microbe mediated process. So when we are going to manipulate the microbes for the production of products, that is a curd, bread and then wine. So they are all comes under biotechnology products. So it is a form of a biotechnology. Next one is a genetically modified organisms. So genetically modified organism means in a particular organism we are going to modify the genetic composition. So that is going to be called as a genetically modified organisms. So these organisms has a restricted sense in the biotechnology field because this GMO is going to be produced by the biotechnology process. But even though it is a biotechnology product, which is going to have a some restricted, because when we are going to alter a normal genome which can able to act vigorously, sometimes it can be useful, sometimes it can be a very dangerous one. So that is why they have a restricted sense in the GMOs only. So some of the techniques which comes under a part of a biotechnology is in vitro fertilization. So when we did the in vitro fertilization, the upcoming will be the product will be a test tube babies. So we can able to produce a test tube babies by the in vitro fertilization. Then next one, we are going to synthesize a gene and then we can use that gene to develop a vaccine to correct a defective gene. So that process also comes under a part of a biotechnology. So in vitro fertilization and then test tube babies and then development of a vaccine, synthesizing a gene. So these are all comes under a part of a biotechnology. Then the European Federation of Biotechnology, that is the EFB. So kindly focus on the name, that is the European Federation of Biotechnology. So these people, this federation, they gave a definition of a biotechnology. So they try to cover both traditional view and then modern molecular biotechnology. That is, we should be have the traditional view and then modern view. So they gave a definition, that is the integration of natural science and then organisms, cells, parts thereof and molecular analogues for products and then services. So we are going to integrate or manipulate organisms by a natural way or an artificial way, either in a cells or in a parts or in a molecular level. For that we are going to getting a product and then services. So this is the definition which was given by a European Federation of Biotechnology. So we are going to see them what are the basic principles of biotechnology. So in that first one will be a genetic engineering. So what is the genetic engineering? We are going to alter the chemical composition of a DNA and then RNA. So the techniques which is used to alter the DNA and then RNA, they are called as a genetic engineering. So we can able to produce a particular phenotypical characters can be altered. So all the characters which is seen in our body will be controlled by our DNA. So when we try to change the chemical composition or position or the structure of a DNA, that outer appearance also they are going to be changed. So let us take a one example. So we are going to alter a particular DNA and then we are going to introduce into the organism. That is a host organism. Host means we are going to introduce something into a particular organism, they are called as a host organism. So if this host organism, they has a light phenotype, that is a skin color. So we are going to alter the skin color DNA and then we are going to introduce into the host cell. Then after some time, which is going to give the dark phenotype of a organism, which means we are going to change the phenotype of the host organism. So before altering the DNA, that host organism will be in a light color. So when we alter it and then when we are introduce the DNA, which makes this light phenotype into a dark phenotype. So likewise we can able to change the one particular character of a organisms by a genetic engineering process. The next one, maintenance of a sterile ambience in a chemical engineering process. So whatever the chemical engineering process which is taken place in a biotechnology, they should be in a sterile ambience because we are going to alter the DNAs. So we are going to alter the chemicals. So it should be sterile and then it should be free from a microbes. Then only the desired DNA or a gene can be altered. So which enables the growth of only the desired microbes or eukaryotic cells in the large quantities. That is why we are using a sterile environment. So which gives you the manufacture of a biotechnological products, because when we are going to manipulate the desired microbes, then only we can able to get the desired products. Some of the products like antibiotics, vaccines, then enzymes, etc. So these are some of the biotechnological products which can able to prepare from microbes. Then we are going to see about conceptual development of the principles of genetic engineering. So what are the concepts which leads to that principles of genetic engineering? So the first concept will be asexual reproduction and then sexual reproduction. So in that asexual reproduction, that genetic information going to be continuously preserved, whereas in a sexual reproduction, they are going to permit some variation. So they identify that the variation will be happen in a sexual reproduction only. So if we alter a chromosome or a DNA, they can also permit a variation. So this gives them the idea of principles of genetic engineering. The next one is hybridization. Hybridization means we are going to prepare a hybrid by introducing the two different gametes from two different plants. So we can able to produce a hybrids in a plant, we can able to produce a hybrids in the animals. So we can able to combine the characters of two animals. Let us take this is an example of a zebra and then horse. So when we combine this, it becomes a hybrid. So that two genes from one from a horse, one from a zebra, we are going to combine and then we can able to produce a hybrid. So this hybridization techniques which leads to the inclusion and then multiplication of undesirable genes along with the desirable genes. So when we are going to prepare a hybrid, they contain all the characters both the animal has, both the horse and then zebra has. So we need to produce only the desirable characters. So when we try to make a progeny of a desirable characters, we are using a genetic engineering techniques. So that leads to the genetic engineering process to come into the society. So some of the genetic engineering techniques are creation of a recombinant DNA. So we are going to recombine a particular DNA strand. So that's also comes under genetic engineering techniques. Then gene cloning. So we are going to clone a particular gene. So that also comes under genetic engineering techniques. Then gene transfer. So we can able to transfer one gene from one animal to another animal or one organism to the another organism. So these are all comes under techniques of genetic engineering. So overcome this limitation, which allow us to isolate and then introduce only a set of desirable gene into the target organism. So these three techniques can able to overcome the limitation of undesirable gene. So we can able to produce only the desirable gene. So that can be allow us to isolate and then introduce only a set of desirable gene into the target organism. So for that they are introducing this genetic engineering process in the biotechnology field. So next one, there is a one question arise. If we are simply we are going to change the DNA and then when we are going to introduce into the some organisms, what would be the reaction? So do you know that likely fate of a piece of a DNA which is somehow transferred into a alien organism? Alien organism means other organism. That piece of DNA would not be able to multiply itself in the progeny cells of the organism, because which is going to be seen as much simply in the cytoplasm when we are going to introduce simply into the another organism. But when it gets integrated into the genome of a recipient, that is a host organism. So when we are introducing a particular gene into the recipient genome, it may multiply and be inherited along with the host DNA. So they can able to multiply and then they can able to inherit it, because which is going to be attached to the normal genome of a particular recipient. So if normal cell division occurs, means this our gene of interest also they are going to get divided. So then they can able to produce a copies and then they can able to inherit it from a parental organism to the offsprings. So alien piece of a DNA has become a part of a chromosome which has the ability to replicate now. So for that, one important part which is needed is origin of replications, which is commonly called as a ORI, that is origin of replication. So if it is going to be any chromosome, when it starts to replicate, they need a origin of replication region. So only if that origin of replication region will be seen, there only that replication going to starts. So let us take this is a one chromosomal circular chromosome. They has a origin ORI, that is origin of replication. So they are going to initiate the replication process. Then they are going to start the replication process and then they can able to produce a second copy. So this is how that replication going to be start when there is a origin of replication region in a particular plasmid or a particular chromosome. So the main region which is important for the replication is ORI. So for the multiplication of any alien piece of DNA in an organism, it needs to be a part of a chromosome. So in that chromosome which has a specific sequence known as origin of replication. So if an alien piece of an DNA which is going to attach with the host DNA, that DNA should has a origin of replication. That chromosome should have a origin of replication. Then only they can able to produce a more copies. So when they start to producing a more replication process, means they are going to produce a clone products, that is they are going to be identically equal. So they are going to all should be looks like an identical. So that is why it is called as a cloning. So we are getting a cloned products by the process of replication. And then we are going to making a multiple identical copies of any DNA template. So we are going to make a multiple identical copies. That is why it is called as a cloned products or a cloned gene. Now we are going to see here how artificially we are going to construct a recombinant DNA molecule. So the two scientists, that is Stanley and then Stanley Cohen and then Herbert Boyer. So what they did means they are try to link a gene which encode an antibiotic resistant gene with a native plasmid of a Salmonella typhimurium. So they took one bacteria, that is a Salmonella typhimurium. So they found out that this bacteria has a plasmid. So what they did means they tried to attach a antibiotic resistant gene along with this plasmid and then they try to remove this plasmid and then introduce into the other cell. So Stanley and then Herbert in 1972, they isolate the antibiotic resistant gene from a other organism, and then they, by cutting a piece of DNA from a plasmid, they found out that some other bacteria which has a antibiotic resistant gene. So this gene will be seen inside a plasmid. So they try to cut the plasmid and then they try to isolate the antibiotic resistant gene. So this plasmid will be responsible for antibiotic resistance in that bacteria. So they are going to cut the plasmid with the help of a restriction enzyme. So we will be studying what is a restriction enzyme. So you just know that when we want to cut the plasmid or cut a DNA, we are using a restriction enzyme, which is also called as a molecular scissors. Why it is called as a molecular scissors? It is an enzyme, so it is a molecule, and then scissors, which is going to cut the DNA or a plasmid. So that is why it is called as a molecular scissors. So we are using a restriction, they use the restriction enzyme to cut the antibiotic resistant gene from that plasmid. Then the cut piece of a DNA was then linked with the plasmid DNA, that is this Salmonella typhimurium plasmid DNA. They used to join this antibiotic resistant gene with the Salmonella typhimurium plasmid. Then this plasmid going to act as a vector, because this plasmid has an antibiotic resistant gene. So we are going to introduce into any other bacteria, they are also going to get that antibiotic resistant gene, so which is acting as a vector, because they are used to carry the one gene from one organism into another organism. So they transfer the piece of DNA which is attached to it. So some of the vectors like mosquitoes, which is used to transfer a malarial parasite into the human body. Then some of the vectors is like a plasmid. So the most important vector is a plasmid. So this plasmid, which is used to deliver a alien piece of DNA into the host organism. So once how they are going to seal that gaps which is present in the plasmid or a gene means, they are using an enzyme which is called as a DNA ligase. So they are using this DNA ligase to link the antibiotic resistant gene with the plasmid vector. So this act on cut DNA molecules and then join their ends. So this DNA ligase which is going to seal the gaps in the DNA strand. So they use a DNA ligase to join the antibiotic resistant gene with the Salmonella typhimurium plasmid. So this plasmid going to act as a vector. So they transfer this into a E. coli, because now this plasmid has a antibiotic resistant gene. So by the restriction enzyme, they cut the antibiotic resistant gene from other bacteria and then try to introduce into the this bacterial plasmid, that is Salmonella typhimurium plasmid. So by the DNA ligase, they are going to join the plasmid and then antibiotic resistant gene. So now this plasmid has a antibiotic resistant gene. So they are going to transfer this plasmid into a E. coli. So why they choose an E. coli means, which is closely related to the Salmonella species. So they can able to replicate using the new host DNA polymerase enzyme. So DNA polymerase enzyme is the main enzyme which is responsible for a replication process. So they can able to replicate inside the host DNA. DNA, that is the E. coli DNA. And then they can able to produce a multiple copies. So they use a restriction enzyme not only to cut the plasmid of other bacteria. They use a restriction enzyme to cut this Salmonella typhimurium plasmid also. So then only they can able to stick the antibiotic resistant gene into this Salmonella typhimurium plasmid. So once they seal this plasmid, they are going to introduce into the E. coli. Then they inside this E. coli, this plasmid going to be replicate, and then they are going to produce more copies. So ability to multiply copies of an antibiotic resistant gene in an E. coli was called as a cloning of antibiotic resistant gene. So we are going to clone an antibiotic resistant gene inside the E. coli with the help of a vector, that is a Salmonella typhimurium plasmid. So the three basic steps which is involved in a genetically modifying an organism. So still now, till now we seen that how to construct an artificial bacteria, which was initially started by two scientists. So likewise, still now we are using a plasmid as a one of a vector. So what are the three basic steps which is used in a genetically modifying organism is identification of a DNA with the desirable gene. So this is the first and then foremost step, because we need to identify the DNA which one has a desirable gene, that is our gene of interest. It is also called as a gene of interest. Then we are going to introduction of the identified DNA into the host cell. So by using a vector we can able to introduce the identified DNA into the host body. Then we need to maintenance of introduced DNA into the host and then transfer the DNA into its progeny. So once it is going to introduce into the host, which is going to get start to replicate, and then they can able to produce a more progeny. So this is the three basic steps which is involved in a genetically modifying an organism. So this is how the GMOs will be produced. Now we are going to see some of the questions from an old question paper. So you till now we learned some concepts. So based upon that concept, the questions will be asked. So you need to answer this all the questions. Plasmid has been used as a vector because both ends shows replication, or it can move between prokaryote and an eukaryotic cell, or it is a circular DNA which has a capacity to join the eukaryotic DNA, or it has an antibiotic resistant gene. The answer is C, because it is a circular DNA. So then only we can able to attach the gene of interest or interested gene DNA. So which has a capacity to join the eukaryotic DNA. So they has a capacity to join the eukaryotic DNA. So that is why we are using a plasmid as a vector. That is the main reason to be used plasmid as a vector. The next question. Extra chromosomal small circular double-stranded DNA molecule in a bacterial cell is surrounded DNA molecule in a bacterial cell is dash. Plastid, plasmid, mitochondria, or a chloroplast. Answer is B, because B is an extra chromosomal small circular double-stranded DNA. So that is which is seen inside the bacteria. So this is a definition of a plasmid, that is which is an extra chromosomal small circular and then double-stranded DNA. The next question. Genetic engineering is possible because we can cut DNA at a specific site by an endonuclease like DNase 1, or restriction endonuclease purified from a bacteria can be used in vitro, or the phenomenon of a transduction in a bacteria is well understood, or we can see DNA by an electron microscope. So which make this possible in a genetic engineering process? Why? Because of what? Answer is B, because genetic engineering is possible because restriction endonuclease, which is going to purify from a bacteria, can be used in vitro. So that is why we can able to introduce the antibiotic resistant gene into the plasmid. So that restriction endonuclease, this is another name for a restriction enzyme. So this restriction endonuclease can be taken from a bacteria, or we can able to introduce into the bacteria, so that can be purified from a bacteria and then can be used in a in vitro stage. So this also this is possible because of genetic engineering. So coming to the summary part, so in this schemes we have learned about introduction about the biotechnology. So what is in biotechnology? So what are the some of the parts of in biotechnology? And then the principles of genetic engineering. So what are the main principles of a genetic engineering? And then what is a plasmid? And then it's important. So plasmid is an extra chromosomal small circular double stranded DNA. So what is it important? Which is going to act as a vector. So that is the importance of a plasmid. So we are going to continue the remaining concepts in the next games. Thank you.